So thank you for the invitation. I'm very happy to be here today and also to talk about Professor Beggs after her wonderful talk. And actually, I'm the first one to introduce the main character of this Congress, which is, of course, the yellow-legged hornet. And in particular, I'll be talking about the lines of research that in the last few years we carried out here at the University of Florence with my research group, and on the biology of Vespa velutina in its invasive range. And just to give a bit of introduction about the invasive alien species, of course, we know that they represent the second cause of biodiversity loss at the global level. And here you can see an array of some of the well-known, the most infamous invaders in different areas and regions, and some of them can have a huge ecological, economic, and public or social impacts, as also Professor Beggs was saying, and often it's very difficult to quantify these impacts. And as we have seen for the yellow jackets, many species of social insects are very good invaders and have a high invasive potential. In fact, we have just seen that, as Professor Beggs was saying, among the Vespids invaders, the 34 species of Vespidae which have been introduced around the world, the 7 most invasive ones are all eusocial and can be ascribed to the genus Vespula, the yellow jackets, as Professor Beggs was saying, but also Polysis paper wasps. And of course, the main characters of this Congress, which are the hornets of the genus Vespa, in particular Vespa velutina nigrithorax here in Europe, and the giant Asian hornet Vespa mandarina, which has been introduced a couple of years ago in the state of Washington in the United States. And but how the invasion starts, and as Professor Beggs was saying, for the Vespila, I mean, in New Zealand, with maybe just a couple of individuals, but is it the same also for probably for Vespa velutina in Europe? And it could be for like most of the invasive social insects, because it is sufficient that one single mated queen is accidentally introduced or transferred into a new area, a new region, and if the foundress, if she encounters favorable environmental conditions suitable for the development of a new colony, she can start a new colony, and in just a matter of weeks or months at most, the colony will be defended by hundreds, if not thousands, of workers. And before the end of the season, the colony will produce a huge number of sexual, which is reproductive individuals that will mate and will contribute to the rapid establishment of the new population and the spread in the nearby areas. And this is probably what has happened with the Vespa velutina nigrithorax and also with other species of social insects, invasive social insects. And so, but who is Vespa velutina nigrithorax? And of course, I mean, it looks almost silly to tell you who is Vespa velutina nigrithorax, because probably all of you already or know just too well who Vespa velutina is. But I introduced this slide not just because it was the first one to talk about Vespa velutina, but also because I would highlight an interesting aspect that many of you probably has already think of. The fact that Vespa velutina is one of the 22 extant species of the hornets, and here we are, I highlighted the 2 species that we have in Europe, Vespa crabro and Vespa orientalis, and the last one in particular is expanding in its native range. In fact, probably due to climate change and global warming, the species is expanding northwards, especially here in Italy. I mean, it arrived in Tuscany, in the southern part of the region last year, and we have just spotted it in Florence a couple of weeks ago at the end of October. And of course, Vespa mandarina, which is invasive in the United States, is not present so far in Europe. But I wanted to point out that as regards the Vespa velutina is native of the southeastern Asia, and it comes with at least 13 subspecies. But interesting thing that the invasive subspecies is Vespa velutina nigrithorax, which has invaded Europe, but also Korea and Japan. And we could ask ourselves why this particular subspecies has become invasive in these different areas and not the other subspecies. I mean, it may be like a random phenomenon, but is it possible that these subspecies could have some, I don't know, preconditions or like pre-adaptations or specific traits that could favor the invasion? And I introduced this slide because these like potential adaptations were investigated by our research group to understand if there are like some specific traits that could favor the invasion of Vespa velutina nigrithorax compared to the other subspecies of Vespa velutina or the other species of hornets. And here of course, I'm showing the colony cycle of Vespa velutina that I assume that all of you already know, so I don't go into I won't go into details because this is the typical colony cycle of temperate social wasps. I mean, the starting of the colony by spring founders is the exit the overwintering diapause, then they produce the first cohorts of workers. But I would like to focus on this part of the colony cycle, which is probably the most interesting one, for the impact that we have on the, of course, beekeeping and the predatory pressure on apiaries, but also the potential impact, for example, of native pollinators and diversity, and also the management cost for the destructions of the huge number of colonies that we can encounter every year in Europe at the end of the summer and during fall, and also because I mean the predatory pressure on apiaries and on native biodiversity of native entomofauna is due to the fact that in this time of the year the colonies have to feed the new generations of sexual larvae inside their nest, and one single nest can produce up to 900 males and 900 reproductive females, which are called giants. And after emergence, these reproductives individual, they will mate. And also this part, I mean, the reproductive biology of the species is quite interesting because the mated giants are the ones who survive the overwintering diapause and then will produce new colonies the following season, and then will help the spread of the invader in the its invasive range. And these are I would say the part of the colony cycle on which we focus with our research, especially for like the reproductive biology and chemical communication, and also for the potential competition during the predatory phase with the native hornet Vespa crabro. But as regards the impact of the Vespa velutina nigrithorax on beekeeping and honeybees, we know that Vespa hornets are often specialized predators of honeybees, and the honeybee is the prey of choice of these of many of the species of Vespa hornets. And in fact, as I said, in the colony cycle, in the months between August and October, we have a very intense predation on apiaries to feed the sexual developing larvae inside the nest. And in fact, in different areas, both in the Asia and in the invasive range in Europe, the honeybee colony losses have been reported between 15 and 30%. And it's almost, I mean, astonishing if we think that a single hornet nest can consume more than 11 kilos of insect biomass in one single season. I mean, think about how many insects, apart from how many honeybees, a single nest of Vespa velutina can kill during a single season. And in fact, as I was saying, the Vespa hornets and Apis honeybees are like the main characters of this coevolutionary arms race. In fact, hornets are perfectly adapted for the hunting on honeybees, and the honeybee is often the prey of choice since it's valuable and abundant due especially to beekeeping activities. And this coevolution arms race, this coevolution between Vespa hornets and honeybees is demonstrated by the consistent number of adaptations and counter-adaptations between predators and prey, as demonstrated by the huge number of studies that have investigated this topic and these fascinating adaptations. And in fact, just recently, we have written with some colleagues this review on this adaptation, and the paper was was accepted just a couple of days ago, actually. And here we give an array of all the behaviors that have been described in different papers. I mean, the fascinating honeybee defense strategies. I mean, from the next shaking to the shimmering display, which is both an intra and interspecific signal between honeybees and hornets, and of course the eavesdropping of the marking pheromone of the hornets by the honeybees, which leads to the well-known hit pollen behavior. And then this very recently described very captivating behavior of Apis cerana, the Asian honeybee, which is able to collect like filth and animal feces and use it to create the spots of this field at the entrance of the nest, which keeps away and repel the hornets of the species, the giant hornet of the species Vespa. But also, of course, the hornets have evolved fascinating strategies to locate and to to catch their prey. For example, the pheromone eavesdropping. In fact, different species of hornets are attracted by, for example, the aggregation pheromone geraniol for honeybees to locate their prey, an honest signal of honeybee presence. And of course, we know all too well the fascinating en masse attack of the giant hornet Vespa mandarina and Vespa soror against the honeybee, and especially Apis mellifera, which has not co-evolved like efficient defenses with the Asian hornets. And then the bee-hawking behavior, which is characteristic of Vespa velutina, with the different hornets continuously hovering in front of the beehives to catch and kill incoming or departing foragers of the honeybee colonies. And especially the predatory pressure of Vespa velutina on the honeybees leads to a considerable impact on honeybees. In fact, we know that the honeybees represent like a huge part of the diet of the Asian hornet in its invasive range, and hundreds of bee foragers can be killed per day by the bee-hawking hornets who besiege the entrance of the colonies in European apiaries. And this, of course, is the cause of a consistent economic impact, also because of the costs linked to the management and the destruction of the colonies, although we still lack precise estimates about the actual economic impact, different from what was shown by Professor Becksford Vespila in New Zealand. But as I was saying, the honeybee is not the only prey for the Asian hornet, and it also preys of native pollinators. And this, of course, cause an ecological impact both on native biodiversity and the key ecosystem service that these pollinators provide, and in turn also on native plant biodiversity potentially. And of course, also in Europe, the social impact, the public health impact due to the venom sting of Vespa velutina and its habit to nest in urban environments or nearby urban environments. And in fact, if we think about, like, surely all of you already have seen these images, but they are, no matter, I mean, they're very frightening. I mean, if we think that, is it possible that one single queen arrived in Europe in the early 2000s, and in like 7 years after the first spotting of Vespa velutina in the department of Lot-et-Garonne, we had 4,000 colonies in France. And if we think about Spain, I mean, in even more frightening, I mean, just 2 nests were spotted in 2012, and more than 10,000 nests were spotted after just 4 years. And the expansion range, the rate is, quite large. I mean, around 100 kilometers per year. But we can also have spotting of nests, even like at hundreds of kilometers from the front of the invasion. And this, for example, was the situation here in some regions in Italy. And this is, alas, the current the distribution of Vespa velutina at the beginning of this year. And as you can see, it's practically all over France, but is rapidly expanding also in northern Spain, Portugal, UK, Belgium, Netherlands, Germany, Italy, and it has been spotted also in Switzerland. So the spread of the species seems like impossible to stop. And that is why, I mean, we could ask, I mean, where do we stand after almost 20 years from the introduction of Vespa velutina in Europe? I very quickly showed that Vespa velutina has different impacts from the ecological, economic, and social point of view. And of course, these impacts have driven like different lines of research on Vespa velutina, with the production of a huge number and an increase of papers, research on this species. But despite the growing attention towards yellow-legged hornet, there are still different aspects of its biology which investigation. In fact, the knowledge on the biology of the species still presents many gaps. And unfortunately, we still lack effective or selective control strategies. I mean, sustainable control strategies in Europe, as also Professor Beggs and Peter were pointing out during like the discussion from the previous talk. And that is why, I mean, we could ask why Vespa velutina is, especially Vespa velutina nigrothorax, I mean, the subspecies which has become invasive in Europe and Korea and Japan, is such a successful invader. I mean, we could ask if, are there any features or traits, as I was saying, like precondition, predation, that could explain, that could have contributed to the success, the invasive success of Vespa velutina? For example, is it possible that Vespa velutina has like higher competitive abilities? Or, for example, higher resistant to pathogens, or the enemy release hypothesis could play a role in this invasive invasion process? Or is it possible that Vespa velutina nigrothorax has a higher investment in reproduction compared to other subspecies or other species of hornets? Or it peculiar reproductive strategies that could favor the spread and the invasion from this subspecies? And that is why is it very important to study the potentially relevant ecological traits and the reproductive biology that could contribute to this, to the success of this invader. And the study of these traits and these aspects of the biology of the species can provide very useful information to explain the invader's success, but also to develop effective and sustainable and selective specific control strategies. For example, the discovery of specific chemicals or attractants for mass trapping, or the selection of selective pathogens, for example, for biological control of the species. And just to investigate, to show the different lines of research that we carried out in Florence, we focused on different unknown aspects of the biology of Vespa velutina in its invasive range. And in particular, we focused on the potential competition with the native European hornet Vespa glabro by comparing especially the diet preferences, the exploratory behavior, and also the individual immunity between the two species. But we also focus on the reproductive biology of Vespa velutina nigrothorax in the invasive range, and the chemical communication linked to the reproductive biology for the, like, for example, specific sex And we also investigated the gut microbiome of both Vespa velutina and Vespa crabro, because we know that the success of many social insects depends on the microbial symbionts that they have in their gut. And so the study of the microbiome of a species could help us understand its ecological role in the ecosystem, but also provide a potential tool for the control and the management of the species. So very quickly into the different lines of research that we carried out. I mean, this was one of the first papers that we published 3 years ago on the potential competition between Vespa velutina and Vespa crabro in Europe. And we adopted a bottom-up approach. I mean, we carried out laboratory-controlled bioassays to investigate the diet of Vespa velutina and Vespa crabro, and also the exploratory tendencies that the two species have. And this was carried out to investigate the potential ability of foragers of the two species to find and exploit different food sources. And I mean, the experimental setup was very simple. Like for the exploratory behavior assays, we just gave the possibility to foragers to explore different confined arenas. And we also measured the boldness of the foragers from the two species, since they were introduced into an opaque box, I mean, a refuge. They could live, and then they were free to explore different areas inside the experimental arenas. And as you can see, there was absolutely no difference in the exploratory behavior between the two species, both in terms of boldness and exploration of the arena. And as regards instead the food item choice test to investigate the food preferences, even here the protocol was very simple. We presented in a confined space the foragers of the two species with different baits, with different food items. I mean, in this case were proteins, but we also did with sugary items. And as you can see, when we present the foragers with honeybees or meat or fish, it was canned tuna, there was absolutely a very overlapping pattern of response, of attraction toward the different baits. And both species were very attracted by the honeybees that, as I was saying, is the prey of choice for this species of hornet. But they were, I mean, there was very little attraction towards the other items that we presented. And this is probably an interesting information, because it tells us, for example, if we think about toxic baiting that has proven so successful in New Zealand, as Professor Beggs was saying, in Europe, probably this won't be that successful, since both the Vespa velutina and Vespa crabro prefer the honeybees if they have the possibility to choose among the different items. But this toxic baiting could instead have a huge impact on the native species of, for example, yellow jackets of polices, as it's happening in New Zealand. So in this case, I mean, we won't hit like the invader, while we would target the native biodiversity. And also another trait that could help us explaining the success of Vespa velutina from the point of view of competition with native species is the immunity, the individual immunity of Vespa velutina compared to, for example, Vespa crabro. In fact, we know that immunity, like both individual and social immunity, is one key aspect of, for the success of social insect colonies. In fact, we know that, I mean, a social insect nest is a very crowded world, and there are frequent interactions among the nestmates inside the colony. And of course, this increase the risk for disease transmission. And the COVID pandemic that we're living at the moment reminds us of this aspect. And this means that pathogen resistance is a trait that should be favored by selection. And this means that a higher immunocompetence a higher resistance to pathogens could really be a successful trait for the Vespa velutina, and could help us explaining its invasive success. And this may be due both because of the absence of natural enemies for the invaders in the newly invaded range, according to the enemy release hypothesis, or, for example, Vespa velutina could be more immunocompetent than the native Vespa crabro. And to investigate this trait, we carried out another very simple experiment, and the paper was very recently published. And we subjected individuals belonging to different castes of sexes of the two species, Vespa velutina and Vespa crabro, to a bacterial challenge. I mean, the protocol was very simple, as you can see. We collected individuals in the wild belonging to different castes, workers, reproductive giants, males, and also spring foundresses in traps that were set up in the field. And we infected them with a mutant strain of the bacteria E. coli. And 24 hours after infection, we sacrificed the individuals, we dissected them, and we homogenized them for plating. And after incubation overnight, we counted the number of colony-forming units that have grown on each single plate corresponding of a single individual of a specific caste or sex. And the number of colony-forming units in each plate is a measure of the bacterial clearance of each individual, which means the ability of the individual hornet to clear bacteria from their hemolymph, and is an indirect measure of the individual immunocompetence of the individual hornet. And there is were quite interesting because we observed that there was actually an effect of caste and species on the individual antibacterial response, while there was no effect of age on the bacterial clearance of each individual. But probably the most interesting results, as you can see from the graph, is that there was a huge difference between the reproductive females of the invasive Vespa velutina compared to the reproductive females, which is autumn giants and spring foundresses of the hornets. And in fact, as you can see, the reproductive giants and foundresses of Vespa velutina were much more immunocompetent compared to the native species. And this is fundamental because the foundresses are the ones responsible for the funding of colonies, so the spread of the species. And also this is very interesting in a point of view of the biological control, because if the native species is much more vulnerable and susceptible to a specific pathogen, it could also be more susceptible to other pathogens. So if we are thinking about adopting a biocontrol strategy to face Vespa velutina, we should be very careful before introducing the potential agent of control in the environment, because it could specifically target the native species, and especially the reproductive females of the native species, which are much more susceptible. So this is an important and key aspect that we should take into account when we think about the perspective of biological control of Vespa velutina. But also in the phase of biological control, we can think about like specific attractants, like pheromones, for example, the sex pheromones, which have been adopted in all of other invasive insects. But before identifying the specific attractants, it's very important to investigate and to deepen the knowledge on the reproductive biology of the species. And in fact, with this was one of the first papers that I've written on Vespa velutina, and here with some colleagues we investigated the cues that could guide mate choice in the males of Vespa velutina towards the conspecific females. And also here, I mean, the protocol was very simple. It was like an experiment that has been carried out in the laboratory, and we presented males, groups of males, with the females belonging to different castes, which showed different indicators, so with different female parameters, which is like the weight, size, and also the abundance of fat bodies. And of course, these are positively correlated in reproductive females. As we know, giants, in fact, are usually larger and heavier than workers, and they have like more abundant fat bodies in the abdomen because they have to survive the overwinter in diapause, and they will have to found new colonies the following season. And as we expected, I mean, not as unsurprisingly, males of Vespa velutina, they preferred larger and heavier females with more abundant fat bodies as like potential mate. I mean, they were more attracted from the stimulus represented by reproductive females compared to workers. And this happened regardless of nestmanship, and could also explain the hypothesis that has been proposed, for example, in France, when we have like a huge rate of diploid males that could result from the male in nestmanship inside the nest, for example. And why males should prefer like larger females and reproductive females? Because those females are the ones that will, as I said, they will survive overwintering. They found the new colonies. They will produce the new generations of Vespa velutina hornets. And this means that they will provide the males with the highest return in terms of fitness. So it makes sense that they should be more attracted to females that present like this indicator of reproductive caste. And apart from the cues that should guide mate choice, we also investigated the chemical communication that could be at the basis of this observed attraction. And in this case, we started from the work that have been published a couple of years before by Wen and his colleagues on the Yunnan subspecies, non-invasive subspecies Vespa velutina auraria. And in fact, in collaboration with Dr. Wen, we carried out similar experiments in the laboratory with the invasive population of Vespa velutina nigrothorax. And we presented males this time with chemical stimuli that were either venom volatiles or the sternal gland secretion that produced the sex pheromone in the Yunnan population of Vespa velutina auraria. And as already demonstrated in these non-invasive species, we observed that venom volatiles had no attractive action on the males, and in fact there was no the volatile compounds that we found through GCMS analysis between reproductive giants and workers, while males were instead attracted by the sternal gland secretion of females of Vespa velutina nigrothorax. And in fact, we observed that in the sternal gland secretion of reproductive giants, there was the presence of the oxyacids that have been identified as sex pheromones in the non-invasive population of Vespa velutina auraria, which means that also for the invasive population of the subspecies of Vespa velutina nigrothorax, the sex pheromones at the basis of sex attraction are the same which are used by the non-invasive population of Vespa velutina auraria. And this could represent a starting point for the development of mass trapping of males, but these chemicals must still be tested in the field in Europe, and this should be like the next step for research. And finally, to conclude my my talk of this morning, I will focus on the very last line of research on which we focus, which is the investigation and the characterization of the gut microbiome of Vespa velutina and Vespa crabro. Because, as I said, the success of social insects often depends on the symbiosis that these insects have with specific microorganisms. And of course, we can cite the termites for the cellulose digestion. And through this study, that is the first study that we published last year, we provided the first comprehensive question of the gut microbial communities in developmental stages, because we analyze both the larvae, but also newly emerging and also reproductive giants compared to workers. And we also compared the microbiome of the individuals with their nest. I mean, the nest and the environment that they inhabit, to understand if they have like a specific microbial signature in their gut, both from the point of view of the bacterial biota and the microbiota of Vespa velutina and Vespa crabro. And very quickly, what we found is that the microbial composition differ across the developmental stages and castes. In fact, we have like a specific microbial signature in larvae. This signature is largely lost with the pupal stage, probably because they expel their gut, I mean the meconium, at the end of the larval stage before entering like the pupal stage. And then we have like the newly emergent individuals, I mean newly emergent females, which have like a lower amount of microbes inside their gut, but there's a progressive increase in gut microbial richness in adults, also compared to the immature brood, I mean the larvae. And probably the driver of such variation that we observed is due to the different diet that we, of course, have like in the larvae, which need protein, compared to the adult individual, which also feed on sugary solutions. And we observed that both the yeast and bacterial communities switch in frequency and abundance during both ontogeny and also according to the hornet reproductive or phenotype. And also interesting, gut microbial communities poorly mirror the microbial communities which are found in the nest, suggesting that the hornet possess a specific microbial signature. It should be very interesting to observe if this microbial signature is different from the native hornet, because as we know, the hornets, but also, for example, paper wasps, they are like ecological reservoir for yeast, and they also play a role as vectors and disseminators of yeast in the field, and they contribute, for example, to the processes of wine production, which means that, for example, the expansion of the invasive species, if they bring with, I mean, the Vespa velutina hornets, they bring with them different yeast, they could, for example, alter production of the different arrays of wines that we have in Europe. And in fact, I mean, this is preliminary results from this paper, which is in preparation. We analyzed, I mean, we compared, as I said, the bacterial and the fungal communities of Vespa velutina, Vespa crabro, and the prey of election, which is the honeybees. And as you can see, it's interesting to notice that from the point of view of the bacteria, there's a clear separation between the two predators and the prey, while as regards the fungal community, we have like a large overlapping among the different species of predator, while the prey, the honeybee, has a very different pattern of fungi inside its gut. And so to conclude, since I mean the time is running late, I mean the could be like the future directions for the research on Vespa velutina and its invasive range. So we have demonstrated, and also other researchers have demonstrated, that Vespa velutina could compete with native Vespa crabro and other native vespids, and especially it could have an impact on the wild pollinators alongside the managed honeybees, although we still lack the actual an accurate estimate of the actual impact of the Vespa velutina could have on both the managed honeybees and the native biodiversity. And Vespa velutina nigrithorax in its invasive range uses and responds to sex pheromones, which means that we could try and adopt, I mean, to test in the field these pheromones to adopt a sustainable strategy for the biocontrol of the species. And also other processes that could be regulated by chemical communication investigated. And also the functional relationship between hornets and their microbial symbionts should be more thoroughly investigated to understand both the ecological role that the species has on the environment and the potential impact on the ecosystems. And finally, it would be important to investigate the response that Vespa velutina could have to different and specific pathogens in order to select the more promising route for biocontrol of the species, a sustainable control of the species, while not affecting non-target species such as Vespa crabro or the other native vespids. And so to go to the take-home message, I would say that despite the years of research that we have behind us, there are still many gaps that we have to fill in the knowledge of Vespa velutina and in its management. But recent advances and also recent techniques are opening new avenues for both the research on the biology of the species and for the control of Vespa velutina nivithorax. And in fact, different strategies and techniques are available against Vespa velutina, and probably we will hear about these strategies and techniques in these 2 days of the Congress. But probably the first step that we have to take is to deepen the knowledge on the biology of the species. And it's very important to establish a timely and efficient monitoring network to understand how the species spread in the invaded area. And we could use, we should use the rapid and effective techniques that have been developed for nest detection, for example, one for all, like the radio telemetry that was developed by our colleagues and friends from France and UK. And we also need standard and especially safe protocols, both ecologically safe and also for the individuals who have to destroy the nest, like for the destruction of the increasing number of nests that we found every year in Europe. And also it should be very important to test both in the field and in the lab species-specific attractants for the selective control of the species, and also potential agents of control such as bacteria or fungi, or also maybe nematodes that could be used for the biological control of the species in a sustainable perspective. And with this, I would like to thank all the friends and colleagues which have helped me in these years of research on Vespa velutina, both in the academic world and also like the beekeepers, which play a fundamental role in the fight against Vespa velutina. And of course, I would like to thank you, all of you, for your attention and for inviting me to give this talk in this Congress. And with this, I finished, and I hope that I didn't take too much time. So thank you.